Good morning. I'm in the laboratory working today on my microscope, and I wanted to show you some fantastic things that I'm seeing. So I found a beautiful fungus. So I'm going to share my screen here. Okay, so here is the fungus, and I'm going to adjust it so you can see it. It's the big long thing that's coming down the center of the screen right now. So we're going to adjust it so the entire thing is in there. I'm going to adjust my light so we can see it a little bit better. You can see the bacteria moving around, which is pretty great. There's different kinds of bacteria in there. But we're going to focus on the fungus today. So right here, I'm right on top of the fungal hyphae right there. This is a beautiful fungus, and it's a big fungus. So if we find our smallest bacteria in here that's in focus, so we want one that's real black and real small that's in focus. So there's one right there that's kind of going in and out of focus, but he's about the smallest one. So how many of those will fit across here? I'm saying about 4 or 5 of those. So this is like 4 to 5 micrometers wide. And then the length of this, we want to measure the length of this. And then we do mathematical calculation and we figure all this out so we know how much biomass is in here. But we're not going to go through the math today. It's complicated. I'll probably never do that on these types of videos. That's way more advanced than what we're doing here. But I am just making you aware of this is what it looks like. And it's pretty awesome. So I'm gonna focus here a little bit. So this part is focused really good right here. And you can see this septa. So that little hash mark across this fungus, there's a black line in between that helps to hold the fungal hyphae wall open so it doesn't collapse. No, not all fungus have those, but this one does. And when you see these uniform septa, uniform meaning they're in there. So there's one, here's one. I'm going to focus the other way a little bit. Coming down here, I'm looking for more down here. Looks like there might be one right here if I could get it focused just right. Possibly one right there. I can't quite see it. Maybe not. But we do have septa in there, which is pretty great. Those cross walls across the fungal hyphae. The other thing is it's brown, and so that's a good thing. That shows that it's most likely a very beneficial, good fungus. But beneficial fungus comes in all colors, even clear. If it was clear, it'd be the same color as this background, so it'd be gray. But the end of this right here, see how it's cut off at 90-degree angle? It broke off at 90 degrees. This one broke off at 90 degrees. So this is great to see this. So this is in my compost, and this is exactly what you want to see. You want to see this in your compost for sure. This is a really fantastic thing of what you want in your compost when you're looking at it through the microscope. So this is an example of a really great fungus that's in here. So I just wanted to show this to you so that you can see what we're looking for. Now I'm going to move to another field of view. So I just randomly move my slide. It's not completely random. We kind of do a grid pattern across the microscope slide, and we look at 25 samples. So I'm going to focus up and down, and we're going to see what is in this one now. But I thought when I saw that beautiful fungus, I thought, this is the perfect example of what to show people. So I popped the camera on, and we captured this. Now, let's look. So right there, I'm going to focus a tiny bit different. Okay, that's about as good as I can get it. But right there, you can see quite a few bacteria. So I'm going to point to them. So right on top of the fingers of this cursor is a real black thing there, a black one there, and there. Those ones are in focus really well. Here. If you come over here, there's 1, 2, 3, 4, 5, 6. So those ones look really good. You can see other ones that are moving. See this white one right there? White, 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 white. And then there's 3 white ones here, a 4th one there, one there, one there. Those are out of focus. So when you do a bacterial count, we would dilute this a lot so that we can actually count everything on the slide. So right now we have, let me see what I did. Our dilution right now is a 1 to 10 dilution. So we want to dilute this maybe 1 to 50 or 1 to 100, maybe 1 to 200, so that we can manage what is in our field of view so we can see it. And that's how you would do a bacterial count. But here again, we're not really teaching how to do that. We're just showing you some critters. So these are cocci because the little round guys, these are these are aerobic critters. That's a good thing because they are going to live in oxygenated conditions and give us good soil fertility. So that's exactly what we're looking for. Let me go to another field of view real quick and see what we have. Okay, so right in the middle of the screen, there's this thing that looks like a piece of broken glass right there where my cursor is. So I'm right on top of it right now, this broken piece of glass thing. That is a mineral particle, like a piece of sand. So that's what a mineral particle looks like. Now, what kind of mineral is it? I have no idea, because a lot of different minerals look the same. So this would not be a test to see what kind of minerals you have. It's most likely not salt, because salt usually is really ornate. So this is a different type of mineral in here. Let me see if there's anything else we can just point out real quick. There are other critters in here. Fun stuff. Let me go to another field of view. Let's see what we're seeing here. It looks like we have, this is probably an amoeba cyst right there. I'm right on top of him right now. That looks like an amoeba cyst. Let me try to focus a little bit. So a cyst means that it's kind of like an egg. He's in a dormant state. I'm guessing that that's what that is. But it's hard to tell. It could actually be a spore of pollen. I said a spore, not a spore, but a pollen. But it kind of looks like an amoeba cyst. We would have to investigate further to be sure. Just keep moving along here. Let me go way over to the other side of the slide. Okay, so there's something interesting right there. Let me try to get that to the right in the middle of the screen so we can see it a little bit better. When you move this microscope thing, a little bit does a lot, so it's moving way over there. But this looks like an amoeba cyst right here because it has the double wall ring around it. So that's a cystated amoeba right there. And this looks like an old shell of a testate amoeba. Let me see if we can focus in on him. So right here, I don't think he's alive. It looks like the shell's cracked right here. It looks like that was his mouth. But this looks like an old shell of a testate amoeba. But I don't think he's alive and active because there's not a lot happening there. So there's good microbes in here. We're seeing good guys. I don't know. This soil sample well, it's a compost sample, but it came from the very top of my compost where it was a little bit dried out. So I just wanted to see what it looked like. Now we have another fungal hyphae right here. So right here, so this is a different width, which means it's most likely a different species than the other one I saw. In fact, I'm pretty confident in that. But you can see a septa right there, that black cross wall thing. And then there's a septa right there. So this is a fungus. It's broken off at 90 degrees there and there. It's a different color. So doing a soil or compost analysis, we write down the color, we write down the size. So there's a bacteria right on there right now that's pretty much in focus. At least he was when I started talking. And we're going to get like 3 of those across there. So this is 3 micrometers wide. That's an Elaine Ingham trick to measure them that way. Let's go to all the way to the other side of the slide here just for fun. Let's focus in on this aggregate. So I'm on the aggregate. It's that big brown thing in the middle of the screen. So I'm focusing. We're coming up. We're looking at this. So this looks like a piece of plant material. You can kind of see the cells inside that plant material. We don't have it's not focusing perfectly here, but anyway, this looks like a cyst in here, like an amoeba cyst or a flagellate cyst. It's probably an amoeba cyst. It looked like it had two walls. But yeah, this looks like plant material. You can see the long strands here, the xylem and phloem cells moving up and down. That's what it looks like to me. Now it's a brown color, has humates in it. The light brown color and the dark brown colors are different kinds of humates. Like humic acid, fulvic acid, ulnic acids, that kind of stuff. Of course, ulnic acids are clear, so you don't really see those, but you can see them, but not with this. Not with this. Not with what we're doing. Not with our equipment we have here today. So this is super dark chocolate brown right here. So this aggregate is interesting. So I want to focus, and see what we can see. And it's such a big hunk that the light's not going through it, so it's not really focusing to see anything inside there. But as you can see, all these little black guys that are barely moving around in here. So these are the bacteria that are eating all this and creating those dark colors. The fungus helps to create the dark colors too. You don't get the really dark humates unless you have the fungus in the soil. Oh, there's a beautiful aggregate right there. Let's see if he's thin enough to see through. So I haven't really seen any flagellates, but I haven't been looking really closely. And this looks like probably a testate amoeba down here. It might be something else, but I'm trying to focus in right here right now. See if we can see through that aggregate. Anyway, there's a lot going on in this sample, but to get a better representation of this, we need to dig down deeper into the compost pile, because I should be seeing more fungus. I've seen those 2 pieces of fungus, which isn't bad, but I like to see more than that. And I like to see, in 25 fields of view, I like to see 3 or 4 or 5 of them that have fungus. So we're looking for strands. So this right here, classic looking. Let's focus in on this. This is the classic shape, size of a testate amoeba. And I think this brown thing here is a piece of debris, but you can see the opening of the mouth right here. It's round right there. This is the test, or it's like an eggshell. It's a shell that's around the testate amoeba. No, he's not an egg. I wonder if he's alive and active. I can't really tell. You don't really see an amoeba in there. Usually you see the amoeba down here, and then they sucked in. And as they sucked in, the vacuum of sucking in pulled debris inside. And we're not really seeing that. So I would say nobody's home here. So we would not count that in an analysis. See this beautiful square thing over here? Another piece of mineral. That's mineral. And then, I mean, like this thing right here, this looks like a fungal spore. Right there, that's pretty classic of that one. This brown one up here, they look like fungal spores. These are probably cysts, amoeba, flagellate cysts. Amoeba cysts are usually bigger, and they can come in a lot of different shapes and sizes. This is an amoeba cyst right here. See, it has 2 walls. Let me focus in on that. Yeah, that's good. You can see the amoeba inside, but you can see two definite walls around this thing. And sometimes the outer wall is like super ornate. It looks like the crown, a king's crown with all kinds of like it's ornamented, so it's shaped funny. But that's beautiful, classic amoeba cyst right there. Anyway, hopefully this helps you to have the best garden of your life by finding critters in a microscope.